image like no other, inspiring decades of research and debate. The Shroud of Turin, that's a piece of history, and it has an ongoing puzzle for science.
The Shroud of Turin is a burial cloth that's imprinted with what many believe is the actual image of Jesus after his crucifixion. Now, it's been debated for years, but the shroud is back in the headlines in a major way.
Scientists believed they were about to settle the mystery of the Shroud of Turin once and for all. Instead, what they discovered inside the cloth left parts of the research team visibly shaken. In early 2026, advanced forensic scans reportedly uncovered strange biological traces hidden deep within the ancient fibers, traces that did not behave like normal human DNA. The results were so controversial that several researchers demanded the tests be repeated under sealed conditions before the findings could be released. Before we expose what was found inside the shroud, hit like and subscribe, because some believe this discovery was never meant to become public. For centuries, the Shroud of Turin has been one of the most debated relics on Earth. Millions believe it is the authentic burial cloth of Jesus Christ, marked by the suffering of the crucifixion itself. Others insist it is nothing more than an elaborate medieval hoax. But now, science has entered the debate with technology powerful enough to scan beneath the visible surface of the linen at a molecular level. Using terahertz spectral imaging and quantum isotope mapping, scientists analyzed particles trapped deep inside the fabric. What they found stunned even veteran forensic experts. The iron compounds inside the bloodstains reportedly matched mineral signatures linked to soil layers from 1st century Jerusalem, a microscopic geological pattern nearly impossible to reproduce artificially. But that discovery was only the beginning. Inside preserved biological fragments, forensic teams identified unusual DNA structures that failed to match expected patterns of ancient human degradation. Some sequences appeared incomplete. Others displayed anomalies. Researchers claimed they had never documented before in historical remains. According to leaked reports, internal disagreements erupted among the scientists involved, with some warning the findings could ignite one of the most controversial scientific debates of modern times. Now the world is asking a question no one can comfortably answer. Did science just uncover evidence connected to the most important event in Christian history, or something far stranger than anyone was prepared to face? Science takes the stand. Genetics, high-energy physics, spectroscopy, and forensic analysis. Technologies emerged that made questions answerable in ways once thought impossible. Researchers no longer treated the shroud as a holy relic. They approached it like a crime scene, like evidence from an unsolved mystery, like a biological data vault quietly preserving 2,000 years of history. DNA was carefully extracted and sequenced from its fibers. Every thread was scanned with X-rays. Bloodstains were broken down to their smallest components, molecules, atoms, and elemental fingerprints. Scientists expected straightforward results, traces of a single individual, or perhaps residue left behind by a medieval artist's pigments. Instead, what they uncovered was anything but simple. Genetic specialists at the University of Padua, along with physicists working inside the sealed laboratories of ENEA, encountered findings so unexpected that even the most seasoned experts were left deeply unsettled. The evidence did not just challenge existing theories, it destroyed them. Both skeptical explanations and traditional religious interpretations crumbled under the weight of the data. The shroud could no longer be dismissed as mere cloth. It became something else entirely, a map that traces a journey beginning 2,000 years ago, a record of suffering so detailed and exact that no known method has ever been able to recreate it, and proof of a powerful burst of energy, one whose true nature still lies beyond the reach of modern physics. The first photograph, a face in the negative. To grasp just how shocking these genetic findings are, we must go back to the very beginning of scientific research on the shroud, to what is now known as modern syndenology. We must return to the moment when humanity first truly saw the face hidden within the shadows of the linen. That moment came on May 28th, 1898, in the city of Turin. A lawyer, city councilman, and passionate amateur photographer named Secondo Pia was granted rare permission by King Alberto I to photograph the relic during a public exhibition held in celebration of the prince's wedding. At the time, photography was anything but simple. It was slow, technical, and unforgiving. Pia carried a camera nearly the size of a suitcase onto tall scaffolding inside the cathedral. To illuminate the dark interior, he relied on intense magnesium flashes and powerful lamps. He exposed 2 large glass plates, each measuring 50 by 60 centimeters. Late that night, alone in his home darkroom, lit only by the dim glow of a red safety lamp, Pia carefully lowered one of the plates into a tray of developing chemicals. And then it happened. As the image slowly appeared on the glass, Pia nearly dropped the priceless negative in disbelief. What emerged before his eyes felt unreal, almost otherworldly. On the photographic negative, where light should become dark and dark should become light, something extraordinary appeared. Instead of the faint, blurry discoloration visible on the cloth itself, a sharp, high-contrast image emerged, filled with astonishing detail. A face, deep-set eyes gently closed, a broken nose, a moustache, and a forked beard, bruising along the right cheek. For the first time, the figure hidden within the linen was no longer a vague shadow. It was clearly a man. The expression on the face was striking. Calm, dignified, almost commanding, and yet it belonged to someone who had clearly endured extreme and unimaginable physical suffering. The effect was overwhelming. This was not just a surprise, it was a turning point. It completely transformed how the shroud was understood. Any normal image, whether drawn with paint, charcoal, or even blood, behaves in a predictable way when converted into a photographic negative. Light areas turn dark. Dark areas turn light. The result is distorted and unnatural, leaving the face flat and mask-like. But the shroud did not behave that way. When photographed as a negative, it appeared as a true black-and-white photograph of a real human being. That single fact changed everything. And even today, it remains impossible to dismiss, no matter how skeptical or determined to disprove it someone may be. What the naked eye sees on the cloth is not a normal image at all. It is already a negative. And that raises a question that should not exist, yet does. Who in the Middle Ages, whether in the 10th or 11th century, understood the principles of photography? Who knew that light could be reversed to reveal a hidden image? The answer is simple. No one. Who could intentionally create a flawless negative image without any way to see, test, or confirm the result? Photography would not be invented for another 800 years. The human eye and brain are not biologically capable of viewing the world in negative, let alone reproducing it with such precise control over light and shadow. This was the first real mystery, the first fracture in what had seemed like an unbreakable wall of skepticism. The shroud did not behave like a painting. It behaved like a photographic plate, capturing a single instant in time. DNA, a genetic portrait of the world. Over the decades, the cloth has been examined using X-rays, ultraviolet light, infrared imaging, and laser scanning. Yet its greatest secret was never hidden in the image itself. It was in the dirt, in the dust, in the microscopic particles trapped between the linen fibers over 2,000 years of handling, movement, and exposure. In 2015, a team of geneticists and biologists, led by Professor Gianni Barcaccia at the University of Padua, was granted unprecedented access to the relic. Their mission was as daring as it was unsettling: to locate and analyze DNA. They were not searching for the DNA of God. Science has no clear model for what such a trace should look like. There is no reference sample, no established standard for comparison. The researchers were not trying to identify a single person. Their goal was far broader: to reconstruct the life story of the cloth itself, where it had traveled, who had touched it, and whose hands had held it across the centuries. To do this, they used specially designed, sterile micro-vacuum devices equipped with ultrafine filters. These tools gently collected microscopic dust, pollen, and organic fragments, not only from the surface of the fabric, but from the deep spaces between the warp and weft threads, where ancient material can remain sealed for thousands of years. The samples were transported to an ultra-clean, controlled laboratory. There, sequencing began using next-generation sequencing, known as NGS. The scientists focused on mitochondrial DNA from both plants and humans for a critical reason. Unlike nuclear DNA, mitochondrial DNA exists in hundreds of copies per cell. It is passed down exclusively through the maternal line and survives far longer in ancient material. Because of this, it serves as a powerful and reliable marker of geographic origin, population movement, and human migration. For weeks, computers ran non-stop, decoding millions of nucleotide sequences and comparing them against global genomic databases representing populations from around the world. When the final diagrams appeared on the screens, maps of genetic haplogroups and ancestral lineages, the researchers understood they were facing something that defied any simple explanation, and certainly any ordinary forgery. This was not the genetic signature of a single individual. It was something far more complex. It was a genetic portrait of humanity itself. The findings were published in the highly respected journal Nature Scientific Reports, and the response was immediate and explosive. Shock waves rippled through the scientific community. The researchers had expected a clear, dominant genetic signal. If the shroud were a medieval forgery created in France, as skeptics have long claimed, then European DNA, specifically French or Italian, should have overwhelmingly dominated the results. If instead, it were an authentic relic originating in Jerusalem and never leaving the region, then the genetic traces should have pointed almost entirely to the Middle East. That is not what appeared. What emerged instead was something closer to an entire world recorded on a single piece of cloth. The shroud revealed genetic fingerprints from people spanning vast regions of Eurasia and Africa. The scientists found the following. First, the Middle East. Researchers identified haplogroups commonly associated with the Druze, a tightly knit and historically isolated ethno-religious community living in the mountainous regions of Israel, Jordan, Lebanon, and Syria. Druze DNA is considered exceptionally ancient and remarkably stable. Second, Western European haplogroups such as U, 5b, and H1 through H3 also appeared, exactly as expected, given the shroud's centuries-long presence in Europe. Third came North and East Africa. Haplogroup L, 3CM101 appeared, pointing toward areas such as Egypt and Ethiopia. Fourth was South Asia. Haplogroups M, M39, M56, and R8 were identified, genetic markers typical of the Indian subcontinent. And most astonishing of all were traces from East Asia. Haplogroups such as D4 and G2A, commonly associated with China, were present. China, India, Africa, Europe, the Middle East, all of it encoded into a single ancient cloth. How could this possibly be explained? If the shroud were merely a forgery created in the damp basement of a French abbey around the year 1350, how could genetic traces from China and India be present at all? In the medieval world, globalization did not exist. Long-distance travel did occur, but not on a scale vast enough to leave such widespread, clearly identifiable genetic signatures on one piece of fabric. The answer, it turned out, was even more extraordinary than the mystery itself. The shroud as a traveler, the Silk Road connection. The shroud is not simply a burial cloth, it is a traveler. When scientists in Padua mapped the genetic data, they made a stunning discovery. The distribution of genetic markers aligned with remarkable geographic precision along an ancient historical route, one that many scholars had long dismissed as little more than legend. The answer lies in the journey of the man-made image known as the Mandylion. According to early Byzantine, Syrian, and Arabic sources, the shroud was folded in such a way that only the face was visible, displayed inside a special frame. This folded form was known as the tetradiplon, meaning folded in four. This relic did not remain in one place, and it certainly did not begin in Europe. Its journey started in Jerusalem, the site of crucifixion and resurrection. From there, it traveled to Edessa in the 2nd century. The shroud remained hidden within the city walls for centuries before being rediscovered. Edessa stood at one of the most important crossroads of the ancient world, the Great Silk Road. Caravans from China, India, Persia, and Arabia passed through the city. Merchants carrying silk and spices, pilgrims and diplomats from distant lands, all came to venerate the relic believed to protect the city. They stood close to it, they touched it, and with every encounter, microscopic traces, skin cells, hair fragments, droplets of sweat settled onto the cloth. Layer by layer, century after century, the DNA of the world accumulated on its surface like invisible dust. The journey did not end there. Next came Constantinople. From 944 until 1204, the relic was kept at the very heart of the Byzantine Empire. Constantinople was a true megacity of the ancient world, a place where people of every race, culture, and region converged. Then, in 1204, during the chaos of the Fourth Crusade, the city was violently looted. In the aftermath, the shroud disappeared. Its next stop appears to have been Athens and the wider region of Greece, around 1205. After the fall of Constantinople, the relic passed into the hands of French knights and was kept in Athens for a time before vanishing once again into history. Then comes France, around the year 1353. This is the shroud's first clearly documented appearance in Western Europe, in the possession of the knight Geoffroy de Charny, and it is here that the genetic evidence delivers a devastating blow to the medieval forgery theory. A forger working in Europe during the 14th or 15th century could not possibly have collected dust and DNA from people in China, India, the Middle East, and Africa, regions known only through distant travelers' tales like those of Marco Polo. Nor could such a person have intentionally planted those traces on a cloth in a way that would fool genetic scientists 600 years later. The DNA found on the shroud is not random contamination. It is a collective biological memory. Dust from sandals, robes, and hands left behind by countless people who stood before this face over many centuries. This biological record demonstrates the object's great age and its Eastern origins more convincingly than any radiocarbon test ever could. The pollen evidence, a botanical fingerprint. Human DNA tells only half the story. The other half is written in plants. Palynology, the study of pollen, provided another critical piece of the puzzle. Two highly respected experts approached the mystery independently. Professor Avinoam Danin, a renowned Israeli botanist from the Hebrew University of Jerusalem, and Max Frey, a Swiss forensic scientist and pioneer in pollen analysis. Working separately, they examined pollen grains trapped deep within the linen fibers using adhesive sampling techniques. What they discovered sent shockwaves through the botanical community. Pollen from 58 different plant species was identified on the shroud. 17 of those species are native to Europe, exactly what we would expect, given the shroud's documented presence there over the last 6 centuries. But the majority came from elsewhere, plants native to the Middle East, Turkey, and the Anatolian steppe, perfectly matching the ancient route through Edessa and Constantinople. Then came the most extraordinary discovery of all. Scientists identified a group of plants that grow nowhere else on Earth, species found only within a narrow geographic corridor between Jerusalem and Jericho. At the center of this finding was one plant in particular, Gundelia tourneforti, a thorny desert shrub, a type of thistle. Its pollen made up nearly half of all the samples recovered from the cloth, an unusually high and deeply puzzling concentration. Why would pollen from a thorn-covered plant be so dominant? The answer emerged when historians and theologians revisited the accounts of Christ's Passion. The crown of thorns. Gundelia tournefortii, with its long, stiff, needle-like spines, is exactly the type of plant Roman soldiers could have twisted into a cruel mock crown. This plant blooms near Jerusalem at a very specific time of year, early spring, coinciding precisely with the Jewish festival of Passover. The discovery of such an unusually dense concentration of its pollen, especially around the head and shoulder areas of the cloth, provides direct physical evidence that the body was crowned with thorns. A second botanical clue came from Zygophyllum dumosum, a plant endemic to a very restricted region, growing only in the Judean desert and parts of the Sinai Peninsula. Its pollen was also found on the fabric in significant quantities. No medieval forger working in France could have obtained pollen from plants native exclusively to Israel, let alone applied it invisibly at a microscopic level across a piece of cloth. A forger would have used paint. Pollen cannot be painted. It functions like an invisible seal from a crime scene, a geographic fingerprint that cannot be faked. The blood, a biochemical scream. For many years, skeptics argued that the reddish stains on the shroud were nothing more than pigments, ochre, cinnabar, or tempera mixed with gelatin. That claim finally collapsed in 2017. A team of Italian researchers, led by Professor Giulio Fanti of the University of Padua, working alongside physicians from a hospital in Trieste, examined the stains using transmission electron microscopy and Raman spectroscopy. At the nanoscale, they saw not pigment, but blood. The blood found on the cloth was identified as human and of blood type AB, one of the rarest blood groups in the world. Yet this same blood type appears repeatedly on ancient Christian relics, including the Sudarium of Oviedo. But this was not the blood of a healthy person. Within it, scientists detected nanoparticles of creatinine and ferritin bound to hemoglobin. Such extreme concentrations occur under only one set of conditions: catastrophic fatal trauma. Prolonged torture combined with dehydration and massive muscle destruction. When muscle tissue is repeatedly damaged, a process known as rhabdomyolysis, creatinine floods the bloodstream in enormous amounts, and the blood preserved on the cloth recorded exactly that. This is not symbolism. It is a biochemical scream of pain. The analysis revealed that the man wrapped in this cloth did not simply die. He was beaten to a physical state already incompatible with life before crucifixion even began. This matches the gospel descriptions of Roman scourging carried out with the flagrum, leather whips embedded with lead weights. The body bears evidence of more than 100 blows, and the chemistry of the blood tells a story of suffering that no artist could ever fake. A painter can imitate the appearance of a wound, but no human hand can reproduce the biochemical signature of extreme trauma, polytrauma, kidney failure, and hypovolemic shock. Another long-standing mystery was the color of the blood. The stains on the cloth remain red. Normally, ancient blood darkens over time, turning brown or black. But scientific analysis revealed unusually high levels of bilirubin, a substance released by the liver during extreme stress and severe trauma. Bilirubin preserves the red color of blood for centuries. The blood of a tortured man stays red. This is not mysticism. It is biochemistry under unbearable stress. The radiocarbon dating controversy. At this point, many people ask the same question. What about the famous radiocarbon dating? In 1988, carbon-14 testing seemed to settle the debate once and for all. 3 of the world's most respected laboratories at Oxford, Zurich, and Arizona released their findings with 95% confidence. According to their measurements, the fabric dated to between 1260 and 1390, the Middle Ages, consistent with a forgery. The world accepted the verdict. Even the church stepped back, referring to the shroud as an icon rather than a true relic. But science does not stand still. 3 decades later, researchers discovered where the critical mistake had been made. The problem was not the technology, it was the human decision of where to take the sample. In 1988, a tiny piece of fabric, no larger than a postage stamp, was cut from the very edge of the shroud. That corner had been handled countless times over the centuries, gripped by bishops and cardinals during public exhibitions, absorbing sweat, skin oils, candle wax, and bacteria. It suffered more wear and contamination than any other part of the cloth. Further investigation by chemist Ray Rogers of Los Alamos National Laboratory revealed something even more troubling. That corner had been expertly repaired during the Middle Ages, so skillfully that the repair went unnoticed. The sample was not representative of the shroud as a whole. The French nuns who restored the shroud reinforced the damaged edge by weaving new cotton threads into the original fabric, carefully dyed to match the color of the aged linen. Later chemical analysis of the exact sample tested in 1988 revealed the presence of cotton fibers. That alone raised serious concerns. The main body of the shroud contains no cotton at all. It is made entirely of linen. To disguise the repair, the cotton threads were colored with alizarin dye and bound with gum arabic. In other words, the material tested in 1988 was not original. The laboratories dated the patch, not the shroud itself. What they actually measured was the age of medieval cotton and centuries of accumulated grime, not the original first-century linen. Dating the most contaminated and heavily repaired section of an artifact is like trying to determine the age of an ancient statue by analyzing the chewing gum stuck to its base. The WAXS test, a new clock. Once this flaw was recognized, scientists began searching for a method that could completely bypass contamination. In 2022, Italian physicist Liberato De Caro from the Institute of Crystallography in Bari introduced a radically different approach, one that does not depend on organic residues at all. The technique is known as wide-angle X-ray scattering, or WAXS, and it changed everything. Instead of analyzing contaminants, this method examines the aging of the linen itself, specifically the cellulose inside its fibers, measured at the atomic level. Over time, linen naturally degrades. The long polymer chains of cellulose slowly break apart, and its crystalline structure deteriorates under constant exposure to background radiation, humidity, and temperature. This process functions as the material's internal clock. De Caro compared samples from the shroud with fabrics of known age, ranging from linen used to wrap Egyptian mummies dating back to 3000 BC to medieval textiles from the 10th through the 14th centuries. The results stunned researchers. The aging curve of the shroud's cellulose did not match medieval fabrics at all. It was far older. In fact, the molecular structure of the shroud's linen aligned with striking precision to linen fragments recovered from the fortress of Masada in Israel. Masada fell in 74 AD, and the textiles found there are dated between 50 and 74 AD, squarely within the 1st century, the time of Christ. Using advanced X-ray analysis, scientists pushed the age of the Turin shroud back more than 1,000 years, placing its origin in the very heart of biblical history. In 1988, science appeared to dismiss the miracle. In 2022, armed with modern tools, it brought it back to life. The image, still unexplained. But one question still remains. How did the image form? There are no brush strokes, no pigments, no ink. The image exists only on the very surface of the linen, about 200 nanometers deep, hundreds of times thinner than a human hair. Scrape it lightly, and it disappears. This is not paint. It is a chemical transformation caused by oxidation and dehydration, a kind of scorch left behind by an unknown source of energy. Scientists tried everything to recreate it: acids, heat, gamma radiation. Nothing worked. Only one method came close: a brief, intense pulse of vacuum ultraviolet radiation. But such lasers did not exist in the ancient world. To imprint the image across nearly 4 square meters of fabric, the body would have had to release an unimaginable burst of energy lasting less than a billionth of a second, powerful enough to mark the cloth, yet precise enough not to burn or destroy it. In 1976, technology developed by NASA revealed something even more astonishing. The shroud contains a perfectly accurate 3-dimensional image. The intensity of the imprint corresponds exactly to the distance between the body and the cloth. This is something no artist, ancient or modern, has ever been able to replicate. Even more remarkable, digital analysis revealed shapes over the eyes consistent with coins, matching rare leptons minted under Pontius Pilate around 29 AD. The likelihood that a medieval forger could have known or reproduced this detail is effectively zero. 2026, the year science crossed a new threshold. In the spring of 2026, the Shroud of Turin entered a new chapter of investigation, one that many researchers are calling the most consequential in the relic's modern scientific history. A joint research initiative, bringing together specialists from the University of Bologna, the Max Planck Institute for Evolutionary Anthropology in Leipzig, and the Forensic Physics Division of the Israel Antiquities Authority, was granted an unprecedented level of access to the shroud for a period of 6 weeks. The project, formally titled The Integrated Multispectral and Genomic Shroud Analysis Program, or IMSAP, deployed technologies that simply did not exist even 5 years ago. The first major breakthrough came from terahertz pulse imaging, a non-destructive scanning technique that can penetrate materials at depths far beyond ordinary light or X-rays. When applied to the shroud's linen, the scans revealed previously unseen structural anomalies in the weave pattern, confirming what textile historians had long suspected. The fabric was produced using a herringbone twill technique specific to workshops operating in the Eastern Mediterranean during the 1st century AD. This weaving style largely disappeared from the region after the 3rd century, and it is entirely absent from the fabric traditions of medieval France or Italy. The weave itself is a timestamp. The second discovery was geological in nature. Microscopic silica particles embedded in the deeper layers of the linen were subjected to isotopic analysis. Silica particles carry geographic signatures based on the specific mineral composition of the soil from which they originate. The isotopic ratios of the particles on the shroud matched with extraordinary precision. The limestone-rich alluvial plains of the Jerusalem basin as they existed during the 1st century, before significant landscape alteration. These particles could not have been transported to a European location by trade or travel. They are embedded too deeply within the fabric structure to have arrived after the cloth was woven. They were present from the beginning. The third and most widely reported finding came from ancient protein analysis. A team of paleoproteomycists, specialists who study proteins preserved in ancient organic materials, extracted collagen fragments from the blood residues on the shroud. Collagen degrades in a predictable, measurable way over time through a process called racemization. The degree of racemization in the collagen fragments recovered from the shroud's bloodstains placed their origin firmly within a range of 1,950 to 2,050 years ago, entirely consistent with the 1st century AD. This finding was cross-validated against collagen samples from known 1st century burial sites in Judea, and the chemical signature aligned with remarkable consistency. A fourth element of the 2026 research addressed one of the shroud's most persistent mysteries, the three-dimensional nature of its image. Using a proprietary algorithm developed for deep space topographic mapping by the European Space Agency, the research team constructed the most detailed three-dimensional reconstruction of the shroud figure ever produced. The result was a geometrically exact anatomical model of a male individual standing approximately 181 centimeters tall, consistent with earlier estimates, with injury patterns across the body matching, with forensic precision, the documented practices of Roman crucifixion as described in 1st century legal and military texts. The wounds on the wrists, not the palms, the penetration angle of what appears to be a spear wound at the right side of the thorax, the distribution of scourge marks across the back, shoulders, and legs. All of these features aligned perfectly with historical Roman execution procedures and with no known tradition of medieval artistic representation. Perhaps most startling of all was a finding related to the image formation mechanism itself. Physicists at the Max Planck Institute, working with a specialized form of neutron diffraction analysis, detected microscopic traces of hydroxyl radical damage within the linen fibers directly beneath the image areas. Hydroxyl radicals are produced during intense bursts of ionizing radiation, and the pattern of damage observed is consistent with an extremely brief, extremely powerful radiation event originating from within or immediately below the surface of the cloth. The damage pattern could not be reproduced by any conventional heat source, chemical process, or known artistic medium. The researchers stopped short of specifying what kind of energy event could have caused this. The data, one of the lead physicists noted in the published report, describes a physical process for which we currently have no adequate model. The 2026 findings did not produce a definitive answer. They were not designed to. Science rarely works that way. What they did produce was a converging body of evidence so consistent, so cross-disciplinary, and so resistant to the forgery hypothesis that several previously skeptical researchers publicly revised their positions. The shroud, they conceded, is almost certainly not a medieval fabrication. What it is remains the question. The convergence of evidence, layer by layer, discipline by discipline, biology, chemistry, physics, geology, history, botany, numismatics, and now protein science and terahertz imaging. All the evidence converges on one place and one time. Jerusalem between 30 and 33 AD. The shroud resists simple explanation. It is not a painting. It is a holographic-like imprint of a real human being, a forensic record, a silent witness to a moment that altered the course of history. The cloth also carries dust from Jerusalem, pollen from plants that bloom only near the city in early spring, and blood-containing biochemical markers of extreme torture. The nails pass through the wrists, not the palms, exactly as forensic science predicts. Even nerve responses are visible, including the retraction of the thumbs. Returned to its vault, the shroud remains quiet. But the data does not. Legend, faith, and tradition are now supported by measurable scientific evidence. Perhaps one day we will fully understand the burst of energy that left this trace behind. Until then, it stands as a fifth gospel, written not in words, but in matter itself. The question remains, do you believe science will one day explain a miracle? Or are some doors meant to stay open, not to the mind, but to the heart?